In this presentation, we're going to be talking about the development of the urinary system, particularly focusing on the development of the kidney. After this lecture, you should be able to describe and define the 3 embryonic kidneys, understand their fate, recall the origin of the cells that develop into the different parts of the kidney tubule, and discuss the differences between the collecting tubule and the nephron in terms of the origin of the cells that give rise to these structures. If we recall, the kidney is a complex organ, and it contains nephrons, of which there are at least 10,000 cells in each nephron. And a nephron has at least 12 cell types. In addition to that, there are 1 to 3 million collecting tubules in a kidney. And so putting the collecting tubule together with the nephron to form the functional uriniferous tubule is what we're going to discuss in this embryology lecture today. To have that discussion, we've got to talk about the development of the pronephros, the mesonephros, the metanephros, and then talk about how the metanephros gives rise to the kidneys. Early in development, about day 22, we can identify the intermediate mesoderm. It is lateral to the paraaxial mesoderm, and it will begin to differentiate at about day 22. The intermediate mesoderm is going to give rise to structures that are called nephratomes, here outlined in blue. Another term for them are nephric vesicles, and these begin to appear in the cervical region early in development. Now, as you can recall, in a lot of the developmental processes, we have development that proceeds in a cephalocaudal direction, and the same holds true for the kidney. So we first see these nephratomes in the cervical region, and then in the thoracic region. And when we see them early on, we will call the collection of nephratomes the pronephros. These early vesicles are non-functional and basically disappear within a few days. As they continue to develop in a caudal direction, the nephratomes will connect with branches off the aorta, and these branches that come off the aorta will give rise to the glomeruli. By day 23, the next set of nephratomes is beginning to develop. The first set, the pronephros, is in orange, and then by day 23 we start to get these nephratomes in the thoracic region, and again they're just developing structures in the intermediate mesoderm, lateral to the somite, and these structures in blue would be called the mesonephros. Finally, later in development, we'll have some more of these structures developing, and that will be the metanephros. Again, the nephric tubules develop in the intermediate mesoderm, and they have now some additional structures we can talk about. They have a Bowman's capsule. That Bowman's capsule will envelop the glomerulus, which arises from the dorsal aorta, and these nephric vesicles will come to join one another distally in a region called the mesonephric duct. The mesonephric duct then will connect segments of nephric tubules. Here we can see in blue this mesonephric duct as it's going to connect the different tubules, which you can see here, which empty into it. In pink then we have the mesonephros, in blue the mesonephric duct, and in green the developing gonad, and we'll talk about that in the next lecture. In purple here is the paramesonephric duct, which again we'll talk about in the next lecture. Today we're going to be focusing then on the mesonephric duct and the nephric tubules. The mesonephric ducts appear at about 24 days. They grow in a cephalocaudal direction, and they grow towards the cloaca. You remember the cloaca is that enlarged end of the hindgut that receives the atlantois. And by day 26, this mesonephric duct is going to reach the cloaca. As it reaches the cloaca, the mesonephric duct is going to develop a lumen and will be functional for a short period of time. At about the end of the 4th week, a short bud appears. It appears at about 28 days, and it is the ureteric bud. The ureteric bud is going to connect to the metanephric blastema during development. And there will be an epithelial mesenchymal interaction that occurs that we're going to talk about in a moment. So basically what we have at this point then at about 28 days is a mesonephros and a mesonephric duct. And between 6 weeks and 10 weeks, that mesonephric duct is actually functional and will produce urine, which will empty into the cloaca. Then, depending on the fate of the mesonephric duct, it will either remain and become part of the male genital duct system, or it will regress in females. So now we want to discuss the development of the kidney, and to have that discussion we need to talk about this mesonephric duct, which also has the name the Wolffian duct. And here we see the Wolffian duct, and what we're going to talk about is the mesonephric duct. It's giving rise to the ureteric bud and the interaction of that ureteric bud with the mesenchyme, which is here called the metanephric mesenchyme or the metanephrogenic blastema. In addition to talking about the development of the nephron, the collecting system, we also need to talk about the development of the vasculature, because blood vessels will have to invade this tissue as capillary networks. When we talk about the development of the kidney, then we can talk about several different processes that occur. When we talk about the interaction of the ureteric bud and the metanephrogenic mesenchyme, we're talking about epithelial mesenchymal interactions. Now, the first thing you're going to say is, well, we have just intermediate mesoderm here. And that's correct. So we're also going to need to talk about how we can get a transformation of a mesenchyme into an epithelium. That also occurs in the development of the kidney. So we're going to see both mesenchymal to epithelial transformations, as well as then epithelial mesenchymal interactions. Both hallmark events in development. So we're going to talk about the first set of events that occurs, which is the interaction of that metanephric mesenchyme with the mesonephric duct. And how the mesenchyme is going to be important for the duct formation. The metanephrogenic mesenchyme is forming in the posterior intermediate mesoderm, and that's what we see here. Here's the mesonephric duct in this little cartoon. Now, how does that intermediate mesoderm form? Well, there are different factors which are important in that formation, one of which, a set of those are the PAX genes. And in addition to that, we have a gradient of bone morphogenetic protein, lowest in the region of the neural tube, if you remember, highest out laterally. And it is in that intermediate region that the intermediate mesoderm develops, so that at some gradient level, both the PAX genes and the amount of bone morphogenetic protein define this tissue as intermediate mesoderm. The actual gradient itself is not known. In addition, retinoic acid and activin play a role in helping to define the intermediate mesoderm. Now, the intermediate mesoderm, once that begins to develop and differentiate, is going to induce the ureteric bud to form. And so here is a mesenchymal epithelial interaction. So the mesenchymal cells then induce the formation of the ureteric bud. One of the factors that's important early in development is the Wilms tumor gene. WT1 is a gene that encodes for a zinc finger protein, and it is requisite for kidney formation and gonad formation. If there is a knockout of the WT1 gene, then kidneys do not form and gonads don't form. And one of the ways this appears to happen is that without WT1, the mesenchyme cannot induce the ureteric bud. In addition, we're going to have other factors such as receptors on the ureteric bud tissue, the tyrosine kinase receptor, RET, and also there are going to be factors made by the mesenchyme such as glial-derived neurotrophic factor, and we're going to see an interaction of these factors which also will cause the induction of the ureteric bud. When we look at the ureteric bud, it's going to give rise to the ureter, and then the renal pelvis, and then the major calyces, the minor calyces, and the collecting tubules. So this is going to branch significantly. One of the things that we can see here is the regulation of this ureteric bud formation and some of the factors involved. Again, PAX2 is important. And here you can see the interaction between the glial-derived neurotrophic factor and RET, and how bone morphogenetic protein can inhibit this if it's too high a gradient. And so just the right levels allow for this interaction, which allows for the development from the mesonephric duct of the ureteric bud. And that ureteric bud then will begin to divide. The ureteric bud not only divides, but then it has a reciprocal induction upon the mesenchyme. And it's going to cause that mesenchyme to condense around each of the branches. Some of that is a result of fibroblast growth factor being produced by the ureteric bud, as well as bone morphogenetic protein 7. One of the things that these do is they block apoptosis or program cell death, and they stimulate proliferation. This is important for this mesenchyme to be able to convert. Without these factors, that mesenchyme is going to die. The mesenchyme then is going to differentiate into nephrons. And what we can see here is first an S-shaped tubule is going to form, and is then going to form around some endothelial cells that will migrate in, and it will differentiate into the nephron. As we look at this, we can recall the complex structure of the nephron with Bowman's capsule forming, with podocytes, and then the proximal convoluted tubule, the Henle's loop, distal convoluted tubule, all being derived from this metanephrogenic mesenchyme. Again, PAX2 is important in inducing the mesenchyme to develop into the nephron, and if PAX2 is not present, that mesenchyme will not condense. And the Wnt genes are also involved in this process. In addition, this mesenchyme not only has to condense but be converted to an epithelium, and so there's an epithelial conversion. And what this means is that we will have a connective tissue morphology to the mesenchyme before the induction. That is, what do you see in connective tissue? Type 1 and type 3 collagen, fibronectin, etc. And after induction, you're going to see things that are reminiscent of the basal lamina. Things like type 4 collagen and laminin. In addition to that, we're going to have things like syndecan and the E-cadherins, which are also important in developing epithelial tissue. Finally, we can talk about these transitions to epithelia, to an epithelium, as being under the influence of the Wnt genes, as we've said, under PAX2, and the cadherins are also going to be expressed as we find those in epithelial cells, not in mesenchymal cells. So the nephrons then are going to develop, and what they're going to then do is they're going to allow for further branching of the ureteric bud. As I said, the ureteric bud then is going to develop into the renal pelvis, the calyces, and then the collecting tubules. So the ureteric bud is going to continue to branch, and branching is one of those processes that we've talked about in several places, and here PAX2 is again going to be important in allowing the branching to occur at the appropriate place. So we have to have the ureteric bud branching appropriately to form the collecting tubules. So we can see now that when we look at the basement membrane of the glomerulus and the podocytes, these now have that epithelial characteristic to them. They have the appropriate junctions. They have the appropriate basal lamina because they've converted now to an epithelial morphology. When we talk about the junctions, connexin 43 is a gap junction protein, and it connects the cells of the nephron. And then PAX2 has to be turned off so that these cells will differentiate. We can also look at the development of the nephron, and we can talk about the fact that Notch is going to play a role in the proximal distal differentiation of the nephron. So especially in the proximal part of the nephron, Bowman's capsule, the proximal convoluted tubule, Notch is important. LIM1 has to be expressed for the renal vesicle to form, and LIM1 then is important in BRN expression And the BRN is going to be important in the formation of the loop of Henle and the distal part of the convoluted tubule. So you can see how we have differential expression of different genes as you go through the nephron, from Bowman's capsule all the way to the distal convoluted tubule. In addition to the glomerulus forming, we need endothelial cells to be recruited. As we said, we need a glomerulus to form. The podocytes, we've talked about how they differentiate into their foot processes and the slit diaphragms, and we've also talked about the glomerular basement membrane now having an epithelial characteristic. So we can now see the true characteristics of the kidney as it begins to form, and we do want to talk about these fenestrated endothelial cells. If we look at the glomerulus, there's the endothelial cells, mesangial cells, the podocytes, and then the parietal cells of Bowman's capsule. And so, how do each of these cell types develop? The Wilms tumor, we said, is important. POD is important. Podocin, GLEP1, and nephrin, these are all important in the signaling pathways that lead to the podocytes. VEGF is secreted by podocytes, and VEGF is important in endothelial cell recruitment and formation into blood vessels. PDGF, platelet-derived growth factor, is important in stimulating some of these cells to become mesangial cells. So you can see that there are a host of factors that are differentially expressed that can help in developing these different cell types. In addition, as we said, we have the connective tissue matrix before, that is type 1 and 3 collagen and fibronectin, and then as the nephron develops, we see laminin and type 4 collagen being expressed, and this type 4 collagen is actually going to shift in its morphology and its types of structures that occur, and this is regulated by LMX1B. As I said, nephrin, podocin, CD2AP, and NEF1 are going to be important in maintaining these foot processes and the filtration slits that you see here. And they're also going to be important in maintaining the basement membrane of the nephron in between that podocyte and the endothelial cells. So you can see that the process is a complex one in which we take two cell types, the metanephrogenic mesenchymal cells and the ureteric bud cells. We have them interact. We have the ureteric bud cells continue to divide and split and branch. Along with that, we have vesicle formation. The vesicle is going to turn into this S-shaped type of structure, which is then going to give rise to the mature nephron and its different components. There are lots of different factors that are important being secreted and made by both the metanephrogenic mesenchymal cells and by the cells of the ureteric bud.